after next week, after we take lecture exam 1. Because the things I'm going to be talking about today, I will be referencing just about every lecture for lecture exam 2 material. Every lecture. This is foundational stuff today. So do not veg. Don't space. Don't be looking at video games. This is a crucial lecture for the rest of lecture exam 2. So take good notes, put it away, take lecture exam 1 next week, then we'll hit it hard in lecture exam 2 after week 5. Again, I'm going at a pretty brisk pace because we lose 2 holidays for your class, and I always struggle with this between spring and fall, but you guys get the same content. Not only are you guys getting the same content as the fall class, you get the same content of every live class I've ever taught. ERT is not diminishing anything in what I'm teaching, okay? All right, so last time we ended up on the slide on ion concentrations. We just basically did an anatomy review. We're going to be on LP pages 19 through 24. And on page 19, we did cover the neuroglial cells, both in the PNS, there's just one, and then in the CNS. All right, now I'm going to have to do this here. And this is going to shrink. This is the only problem I have with this system. There we go. Okay. And this was actually one of the essays for lecture exam 1, and that is the different ions between ECF and ICF. I want to highlight some things that contribute to what we call membrane potential. And we had this in the ECF for lecture exam 1. In the ICF, there are some other ones as well for lecture exam 1. But what's really key for the development of a voltage difference across the plasma membrane is that there's more positive potassium inside the cell, but there are many more positive ions outside the cell. And even though there is more chloride outside the cell, we have these things called organic anions. Remember, organic means it has carbon in it. While when we talked about ions or electrolytes, there's actually 2 subcategories. And those are cations, which are positive, and anions, which are negative. Okay. Now, when we refer to organic anions, we are basically referring to proteins. Okay, and proteins are negatively charged. So those are the organic anions. So you have more positive ions outside, more negative ions inside, and that causes there to be a voltage difference across the membrane. And I mentioned this on Monday lecture, and we're going to start there. Membrane potential is the voltage difference across a membrane. We actually have that in lecture exam 1. Can anybody tell me where we actually have talked about a membrane potential in lecture exam 1? And I'll give you a hint, it's under metabolism and the 3 stages. I'll give you another hint. It's in the ETS, electron transport system. I'll give you another hint. It's really explained in the chemiosmotic theory.
Positive feedback loop?
Okay, good try. Think of specifically ATP metabolism, 3 phases or 3 stages, the last one being the ETS, the big payoff. Chemiosmotic theory. Whew, shit, you guys got some studying to do. All right. The proton pumps. As the electrons are being passed from boulder to boulder, they are pumping protons, which are positive cations, free hydrogen ions, into the intermembrane space. Does this ring a bell? This causes there to be a lot of potential energy stored in a pH and voltage gradient. Do those words ring a bell? Look it up on your essays on chemiosmotic theory. Voltage gradient is this. Remember the word gradient, you can translate to difference. Now, does that ring a bell? There actually is a chart. So last week, or last lecture on Monday, I said plasma membrane. And that's really what we're talking about with neurons. But it can be across any membrane, including the inner mitochondrial membrane. That's on the chemiosmotic theory. Okay? All right. Okay, you guys are freaking me out here a little bit. All the blank stares when I was asking that question. Okay, now we got to go on. Okay, let's first talk about nerve fiber characteristics. When we talk about a nerve fiber, and I put up multipolar neurons up on the whiteboard on Monday, we're talking about the axon. So right there on the very top of the slide where it says nerve fiber, just say equals axon. They show these characteristics: excitability or irritability, and this is a characteristic response to stimulus. One of the ways they respond is an action potential, and action potential is using the same word. In fact, that'll be number 2, because there's a whole different list. There's a whole bunch of different membrane potentials. I just have resting membrane potential because that's the one I'm going to start with first. Action potential will be the second. This is what some of you may have referred to as nerve impulses in the past, and you want to get away from calling them nerve impulses, except for when you're talking to patients. When you're talking to patients, call them nerve impulses. When you're talking to colleagues, call them action potentials. Why? Both neurons and muscle cells carry them, and muscles are not nerves. Okay? Now, there's 2 things that dictates the velocity of an action potential. What you might want to write next to action potential, it is not electricity. Electricity going through the copper wiring in your house or your car is going at near speed of light. And action potentials are not electricity. They are way slower. Action potentials are not electrical waves, like the copper wiring in your car. They are chemical waves. That's why they are much slower than electricity. And the speed of an action potential varies from muscle cell to muscle cell and neuron to neuron. And for neurons, there are 2 things that influence the speed of an action potential. One is axon diameter, and this is on the bottom of page 19 of your lecture packet. The wider the axon, the faster the action potential. And here's a figure right here of a cross-section through a nerve trunk. Actually, this is one fascicle in a nerve trunk. So this is perineurium, if you guys remember that from anatomy. Perineurium surrounding a fascicle. All these little dots inside are cross sections of axons. And the big green ones are fast, and the tiny hollow ones are slow. And the relationship is linear. Look at that graph. This, by the way, we will talk about later in circulatory physiology. This is a proportional relationship. Whenever you have a line going in this direction, this is a proportional relationship. You increase diameter, you increase velocity, and the line goes in that direction. Proportional relationship. The other thing that influences is myelination. Hopefully this was covered in your anatomy class. Those who took me for anatomy class, I referred to that last semester as in the live class. I get everyone out of their seats in the whale room. We form a ring around the room and we do the wave, because that's exactly what the chemical wave is for. It is a wave of an influx of sodium in, down the axon, boom, 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 boom. Well, if you're unmyelinated, you have got to open up and do the wave every student all the way around the whale room. And that takes a while to get around the whale room. But if this Schwann cell in this figure, neurolemmacyte, covers 3 students, that would be 1 millimeter, the action potential actually jumps every 4th student. So the first student will start, boom, 3 students don't do anything. But then the 4th one does it immediately as that first student comes back down. We do this wave where boom, boom. Just do it like this, boom, 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 boom. And as soon as the first student gets back down to horizontal, the second student starts. As soon as that student gets down to horizontal, that student starts. Everybody got it? That would be unmyelinated. All those gates have to be activated. In this, what you're seeing in this figure, what we call saltatory conduction, the first student goes, boom, boom. The next 3 students don't. But the 4th student, as soon as that first student comes back down to horizontal, goes. Next 3 students don't do anything. Now it's the 7th student, boom, boom. And it's jumping from node to node. This saltatory conduction is the action potential jumping from node to node, and that is much faster. This animation should point that out. As it turns out, then if you look at the table on the bottom of page 19, the wider axons tend to be myelinated. They kind of go hand in hand. The smaller axons tend to be unmyelinated. Okay, 2 things that influence the speed of action potentials. Same thing here, look at wider axons tend to be myelinated. This really narrow one is not. This is like temperature and pain. That's why you got to be really careful with temperature. If it's really hot or really cold, you can have tissue damage before you feel that it's hot or cold, because those fibers are really slow. Okay. This is showing you saltatory conduction from your textbook.
Question?
Yep.
Okay, so like using that graph from the previous, or that chart from the previous slide, so it's not only like the size of the axon, but it's the amount of myelination that's on it as well?
Yes.
Okay.
And it's not really the amount, it's whether it is myelinated or not. If it's myelinated, basically what it is that the neurolemmacyte or oligodendrocyte wraps membrane over and over and over and over and over, and it renders those sodium channels that we will talk about in a little bit, renders them moot. And that's why it has to jump from node to node.
Okay, gotcha. So on like, for example, with the first and the third, like group 1 and then 3 on that slide, it's the speed's like dramatically different. Is that because of the ratio of myelination to size of the axon?
It's both.
Okay.
Yeah. And that's what this graph is showing. It's both. All right. So yeah, with unmyelinated fibers, yes, increased axon diameter will slightly increase the speed. But if it's myelinated, you get wider axons and boom, it really increases the speed. Everybody see that in that graph, bottom right? That means this line, the black line, is going up at a much milder line. It's still proportional. Still wider you get, the faster you get, but it's much flatter. If it's myelinated, if it gets wider, it skyrockets in speed. Everybody see that? All right. All right, so let's talk about membrane potentials. Again, we have different ion concentrations between ECF and ICF. We just talked about that. The very first membrane potential we're going to cover is resting membrane potential. Now, every neuron has a slightly different resting membrane potential. They are not all the same. When we talk about after lecture exam 1, we're going to talk about this thing referred to as recruitment. This is relying on this bullet, that different neurons have different resting membrane potentials. I'll just plant a seed right now. We'll really talk about it after lecture exam 1. When it comes to sensory input to your brain, your brains feel nothing. Your brains see nothing. They hear nothing. They taste nothing. They smell nothing. Your brains can't do anything like that. The only thing your brains can do is receive action potential from sensory neurons. It goes to the thalamus. The thalamus redirects it to different lobes of your brain. And if it goes to the back, your brain will generate a visual illusion. If it goes to your parietal lobe, it'll give your brain an illusion of taste or of touch. If it goes to your temporal lobes, you'll generate an illusion of sound. And it's going to generate an illusion of sound even if there is no sound. Your entire existence is a brain-generated illusion. You see nothing. You think you see me in that tiny thumbnail? You think you see this PowerPoint slide? Your brain doesn't see it. Your brain is generating an illusion. Just wrap your brain around that. And then we'll talk about that after lecture exam 1. Oh, but lecture exam 1 is real. Okay, let's go. We're going to use, though, as a typical resting membrane potential, -70. A lot of online sources use -70. Your textbook uses -70. That's what we're going to use, but just know it is typical. It's an average. It is not every neuron has -70 resting membrane potential. Now, the other thing that I want to point out is the word resting is a misnomer. The word resting is a misnomer because this whole thing is actually powered by sodium-potassium pumps. And this is one of the 6 things that are contributing to resting membrane potential, because 3 doesn't equal 2. You guys remember that when I was going over active transport and sodium-potassium pumps? 3 does not equal 2. That is contributing to the -70. Because all cells have it, all cells actually have a membrane potential. But we're only talking about right now neuron membrane potentials. All right, well, what contributes to the -70? And this is what I have on the whiteboard here. I'm about to go over it. Resting membrane potential, 6 things add up to -70. That is the next slide and the slide after that. Slides 34 and 35 are about what adds up to resting membrane potential. Okay? Any questions? Let's do it. And this is also listed on page 20 of your lecture packet. They actually listed it under 7 things, but this makes more sense to me. All right, so we have the ion concentrations. We already covered that. Right here, if you look at this little figure on the top right, is showing you that one thing that makes a big difference is those organic anions. That's what this A minus is. Those are the proteins. Okay. Also, we have protein channels for facilitated diffusion. In fact, for number 2, you should write facilitated diffusion, because if you're going through a protein channel, that's what it is. It's not single word diffusion. But the rates of diffusion are different. And that's also kind of shown here with a little bit of diffusion here. This is like a dashed line. Look at my white arrow. Yeah, a little bit of sodium goes through facilitated diffusion in all the time. A little bit of chloride goes in, but it's fighting against a negative charge, because opposites attract, likes repulse, and if it's more negative inside, yeah, chloride will go in, but it doesn't really want to head towards negative. But look at this arrow right here, it's solid. And that's because a whole lot more potassium leaks in. Leaks out. So much more is going to go in, and so much more of this, so much little of this is going to go in, unless there's an action potential. So there's differential facilitated diffusion rates. Then because of opposites attract, since there are more negative anions inside the cell, more cations outside the cell, and opposites attract, they will get right up against the membrane. So if you notice in this figure, I know it's a little pixelated, but you got this mixture in the ECF, but since the cell is negative, most of those cations will be right up against the membrane. Inside the ICF, you still got a mixture of ions, but because the ECF is positive, those negative anions will be right up against the membrane, because they are attracted to the opposite. Opposites attract. Everybody got that? That actually also increases and adds to the -70. And again, ions cannot go through the lipid bilayer. They are separated. They'll get really close right up against the membrane, but they can't go through unless they have a protein channel. This is actually a statement that is tied to number 2. Way more potassium leaks out than sodium leaks in. And also contributing to it is 3 doesn't equal 2. The sodium potassium pumps by themselves won't generate -70, but it adds. If you look this up online, there'll be some good little websites that show this voltmeter and show these various things. And again, you know, in the lecture packets at 7 and my PowerPoint at 6. But as they add in each one of these features, you'll see the voltmeter going up, 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 up. At the 6th one, boom, it's at -70. So not any one of these 6 will generate -70. It's all of them together results in -70. And that's what I have on the whiteboard. This is slide 35.
I have a question.
Okay.
-70, does that exist in the cytosol but not in the organelles?
We are talking about the general ICF and the cytosol versus the ECF, yes. But there are differences within organelles as well. And the example of the mitochondria with the chemiosmotic theory being an example. Okay. All right. So right now we're talking about just basic ECF versus ICF. Here's your textbook showing you the various forms, various attributes adding up to the -70. Oh, wait a minute. I gotta see if I can get this to work. Hold on just a second, right? Oh, I forgot to do one thing when I set this thing up. I fixed this thing. You know, remember we had that video on mitosis on Monday? I got this thing fixed and I skipped the step. I don't think you guys want me to end the meeting and restart it, do you? Ah, crap. Let me change the view. Let me see if I can do it another way. Okay, well, I actually got this thing working. So I'm going to hope this works because the system, he said that it might remember. Let's just see. It would help if it would play.
The human brain alone contains about 100 billion.
Do you guys hear it?
Yes. A neuron, like every other cell, has positively and negatively charged ions, inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partition of charge creates a voltage difference across the membrane, known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of -70 millivolts. The resting membrane potential depends on two factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neurons than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane. Sodium and potassium ions, leak channels in the plasma membrane, allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable or leaky to potassium ions. These positively charged potassium ions leak out of the neuron. The inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at -90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to -70 millivolts. Sodium potassium pumps activate the ions out of the neuron, and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potentials.
Okay. Everybody got that? Okay, resting membrane potential. Many things coming together to add up to -70. And that is example only. All neurons are different. Okay, now let's talk about the action potential, which is covered on page 21 of your lecture packet. But before we do that, we have to talk about various terms. Next to this slide, write the key to the rest of lecture exam 2. This slide is the key to the rest of lecture exam 2. And these terms, when I say that, this is key for the rest of lecture exam 2. These terms must become as natural to you as breathing. If they are as natural to you as breathing, you're going to be in the ballpark. If you have to think about it, other questions are going to be way too difficult. This must become as natural as breathing. Now, we already talked about how minus 70 is resting membrane potential. So I have a graph on the whiteboard, and it is the same thing as what's on the slide, okay? What you have to know is that there is a higher value above resting membrane potential, and I'm going to draw it at minus 55, and that's threshold. And you can see it in the graph. But before we get there, let's talk about this term polarized, because that is actually the equivalence of this. We will call this polarized. Oh, this blue is kind of wimpy. No, he's purple, I guess. I'll use this. Okay. Now remember when we talked about water and polar molecules, and I said it's polar if you got a north pole different from a south pole. Does that ring a bell with water? Look at this membrane right here. If this is the ECF and this is the ICF, and it's negative on the inside. And you know, when we say minus 70 millivolts, it's minus 70 millivolts on the inside. The exact same resting membrane potential could be called positive 70 millivolts on the outside. That's the exact same thing. Does everybody understand that? It's just a voltage difference across that membrane. The reference for us is on the inside of the cell. Well, if on the inside of the cell it's negative, and the outside of the cell is positive, we have a north pole that is different from a south pole. And that's what we call minus 70 millivolts polarized. Everybody got that? Across that membrane, there's a north pole that's different from a south pole. Here, right here, it's red, positive north pole. Blue, negative south pole. It's polarized. Everybody got that? Again, this must become as natural as breathing. All right, if you get some signals on the neuron, and this is part of that excitability, irritability, you can have some signals onto a neuron that will make it more negative. If it makes it more negative, that is hyperpolarized. Okay? But it's eventually going to return back. Because of all those 6 things, even after that little change, it's going to go back to minus 70. But since to aspire an action potential, we must reach threshold. If it is more negative, hyperpolarization is inhibitory. Why? It's further away from threshold. Okay. If we get closer to threshold, that is less negative on the inside. Minus 70 is polarized. Well, now it's less from minus 70. That is depolarization. And because depolarization is closer, oh, I guess I should do it in orange, is closer to threshold, that is excitatory. And if you get enough depolarizing signals onto that neuron, it's going to reach threshold and fire. So that we consider excitatory, closer to threshold. But if it doesn't reach threshold, it'll just return right back to minus 70. Everybody got it? And the return, oh, it's green. I don't, it's whatever. So if you return to minus 70, that is repolarization, because why? Minus 70 is what?
Polarized.
Polarized. Does everybody get those three terms? These terms must be as natural as breathing. Resting membrane potential is polarized. Why? Because across the membrane, there's a north pole different from a south pole. If you have signals that move away from threshold, it's more negative, hyperpolarized. That's inhibitory, it's further away from threshold. But if it returns to minus 70, that's repolarization. If you have signals that make it less negative, you're closer to threshold, but since it's less negative, it is depolarization. Since it is closer to threshold, it is excitatory. And if you return to minus 70, that is repolarization. Okay? Does everybody get that? Because this is basically what's going to happen. Eventually, if you get enough strong depolarizing excitatory signals, boom, you're going to hit threshold. Boom, it's going to go up and peak at about positive 30. Again, everyone is different. And then it's going to return. I'm going to talk about these channels in just a second. And then actually it overshoots minus 70. Until it comes back to minus 70 eventually. And that, again, sequential thinking helps. And you want to think through an action potential sequentially. But before I do that, this slide must be as natural as breathing. Any questions on that? These terms are all relative to threshold and resting membrane potential.
I have a question.
Okay.
What determines the threshold voltage? Why negative 55 millivolts?
It has to do with the voltage sensitive sodium gates. Does that help?
Okay.
Yeah. Some voltage sensitive sodium gates are so sensitive that maybe at minus 60, they'll start opening. Some other voltage sensitive sodium gates are not as sensitive, and you've got to go up to minus 45 before they'll start opening. Does that help? Okay. Okay, so return to minus 70 is repolarization. I didn't have an arrow, but this is actually right here also repolarization. Any change from minus 70 will be either hyper or D. Every return to minus 70 from that change is repolarization. All right, now let's talk about some of the protein channels, ion channels that play a role in resting membrane potential and action potentials. There's two different types of potassium channels. There's only one type of sodium channel, but the sodium channel does have two different gates in it. All right, the two different potassium channels. This first diamond, I would write open pipes. They're like a wide open pipe. Write that next to this first diamond if you want to understand this quickly. This first one is a wide open pipe. Actually isn't. Just so that you'll understand this, I'm teaching you a lie. Then we have potassium channels that actually have a gate on it. Okay? And these are sensitive to voltages, and they are open or closed depending on the voltage. So these are voltage sensitive potassium gates. See, and this is two kinds of protein channels for potassium, K+. Open pipes and gates, and this is sensitive to voltage. All the sodium channels are gated, and they have two different gates on it. They have a voltage sensitive gate, and they have an inactivation gate. And on these diagrams, one is here, and one is right here, right in the middle of it. These gates are never wide open. They tend to flicker. So even when the conditions are such that they should be wide open, they're not wide open, they're flickering. The gates are kind of like regulating how much sodium is going through. And that is connected to this, number 5. Connect that slide on the sodium gates to number 5. Potassium has two different types of gates. One is permanently open pipes. That's why more potassium leaks out. Sodium is never open. Even when they're supposed to be open, the gates are still flickering, regulating how much sodium can go in. You see how those are connected to number 5 here. Okay? Everybody got it? That's why I put this right here. The permanently open pipes is the reason why way more potassium leaks out than sodium leaks in, because there are no wide open pipes. Even when it's supposed to be open, they're flickering. And that says it right here. Occasionally flicker open and quickly close. That's when they're supposed to be open. Everybody see that? Okay. And again, there is the voltage sensitive gate and the activation gate. All right, so let's talk about action potentials. Again, they are all or nothing, and they are all or nothing based on threshold. And some of these things are talked about on page 21 and 22 in your lecture packet. All or nothing means there are no mini action potentials, there are no partial action potentials. All based on threshold. You hit minus 55, you will have the full form action potential. You don't hit 55, it'll look like this. It'll just return back to minus 70 without a spike. Hit threshold, boom. Don't hit threshold, blant. All or none. Every action potential spike is the same, and on the second diamond, write for that neuron. For that neuron, every spike is the same. different neurons have different spikes. But for that neuron, it's all or nothing, and if it's all for that neuron, it'll be identical. And the timing of it, and this is why this is in milliseconds, is really quick. And so we also refer to action potentials as spike potentials. And remember, for lecture exam 1, so this is part of one of the essays, we tend to see in negative feedback loops small oscillation. We tend to see in positive feedback loops large spikes. And an action potential is a positive feedback loop. And it shows you the large spike. You can see it on the slide. Okay. Again, if it's subthreshold stimulus, and this terms are on page 22, doesn't reach minus 55, goes right back down to threshold, to a resting membrane potential. But if you hit threshold, boom, you'll get the spike. Now, one thing I would write on this slide from your textbook is if you have a supra threshold stimulus, you will have multiple spikes. Everybody got it? And that's an error on the publisher's part. To just draw one action potential is incorrect. There would be multiple spikes really quick. All right, so let's go through the sequence. Sequential thinking helps. First, you have to start at resting membrane potential, and that's why we covered it first. A neuron cannot fire unless they start at resting membrane potential. So it's very important. Then you got to get excitatory stimuli, depolarizations that allow it to reach threshold. And we're using minus 55 as an example only. Again, different neurons have different thresholds. That is going to initiate the positive feedback loop for sodium. And I actually illustrated this when I was talking about positive feedback loops, I think in the very first lecture. I gave you an example of an action potential. There's a positive feedback loop for sodium. These sodium channels are voltage sensitive, and they are different. You reach threshold, some of them are going to open. That allows more sodium to come in. That makes it more positive. And that causes more sodium gates to open up. That allows more sodium to come in, and it gets more positive, which opens up more sodium gates. That allows more sodium to come in, and it gets more positive, which opens up more. Do you guys remember that when I did that first lecture? Positive feedback loops. I use this example, and that's why this orange line is skyrocketing. And that's it right there. So this is the first diamond. You can see this in the thumbnail. This is the second diamond. When it hits plus 30, and again, that's an example, the voltage-sensitive potassium gates will now open. And that is causing potassium to leak out. Now there you want to think more algebraically about this. If you add into the neuron sodium channels, it's going to get more positive. But if you open up potassium channels, because of the concentration gradient, this is all facilitated diffusion, it's got to go from high to low. That means it's got to go from inside the neuron to outside. If you lose positive ions, you get negative. You just got to think it through logically, algebraically. If you sucked at math, this is going to be more difficult. Add positive ions, it gets positive. Lose positive ions, it gets negative, and that's causing the red line. That is repolarization. Now, there are two different types of these sodium gates. So on this slide here, maybe draw a line to the second from the second diamond and write there are fast gated channels and slow gated channels for potassium. There are fast gated channels and slow gated channels. So there's actually two types underneath this diamond. Fast gated channels for potassium and slow gated channels for potassium. These ones here for this third diamond are the, and right next to this third diamond, fast potassium gated channels. On the third diamond, fast gated channels for potassium. And the potassium gates is what ends the positive feedback loop. All of our positive feedback loops in our body, and there are not many, that's the exception, not the rule, all have an endpoint. And you gotta have an endpoint, otherwise you're dead. Well, in the case of the action potential, it's potassium voltage sensitive gates. First, it's the fast ones, and then it's the slow ones. They open kind of late. They open around here, and then they stay open. And because they stay open, it goes past -70. It overshoots -70 down to -80. And again, that's an example only. And that is referred to as the after hyperpolarization. So we got 1, that first diamond, got to hit threshold. 2, you got to have the positive feedback loop. 3, once you hit plus 30, you got to have repolarization from the fast potassium channels. And then 4, you got the slow potassium channels. That makes it overshoot -70. During the spike itself, the sodium potassium pumps, and it's really, I mean, it's only 3 milliseconds, they shut off. But after it gets to the after hyperpolarization, the sodium potassium pumps turn back on. Now this after hyperpolarization time is really crucial when we start talking about refractory periods, because we are now further away from threshold. But when the sodium potassium pumps turn back on, it gets us that. And that's the last part of the sequence. Okay, so let me summarize on the whiteboard here. First, you gotta have a depolarizing signal, probably many signals that are depolarizing, and they all add up to threshold. You hit threshold, boom, it opens up sodium gates. So number 2, the positive feedback loop is about sodium gates. Then once you hit plus 30, you got the fast potassium gates. As it is repolarizing, around in the middle, the slow ones finally activate. And they stay open too long, and it overshoots -70. And then once it overshoots, the sodium potassium pumps turn back on. Does this help connect that to that PowerPoint slide? 1 is getting enough of these signals. Boom. Oops, just erased it. That kicks in the positive feedback loop for sodium gates. That's the positive feedback loop, and that's why it's a spike. Once it hit plus 30, the fast potassium gates open, these close, they inactivate. Boom, it drives it right back down. Slow ones open up around here. They stay open too long. You overshoot for the after hyperpolarization, and then the sodium potassium pumps kick back on and return to -70. Sequential thinking helps. This is the sequence, but this is going to be key for the rest of lecture exam 2 material. And this is what must become as natural to you as breathing. Can I be any more clear? Yeah, I gotta get going here. Okay, your textbook just makes it here, depolarization, repolarization after hyperpolarization. That's what they're doing. But I wanted to tie it to my PowerPoint slide.
By generating and propagating action potentials. Most action potentials originate near the axon hillock of the cell body in the initial segment of the axon. It then travels the entire length of the axon. A closer look reveals that during an action potential, voltage-gated channels open and close, altering the permeability of the plasma membrane to sodium and potassium ions. A threshold stimulus changes the shape of the voltage-gating cell, causing their activation gates to open. This event marks the beginning of phase 1 of the action potential, known as depolarization. As sodium ions diffuse into the axon, the membrane potential becomes less negative. This causes more voltage-gated sodium channels to open, and the membrane potential soars to just 30 millivolts. At this point, two key events occur. The inactivation gates of voltage-gated sodium channels close, and voltage-gated potassium channels. These two events mark the beginning of phase 2 of the action potential, known as repolarization. As potassium ions diffuse out of the membrane potential becomes negative again. However, the membrane potential continues in the negative direction, going beyond the resting state of minus 70 millivolts. This marks the beginning of phase 3 of the action potential, known as hyperpolarization. During this phase, voltage-gated potassium channels close and all voltage-gated sodium channels are released from the activation. Squeeze ions channels open and the membrane potential returns to the resting state of minus 70 millivolts. Polarized foundations are another action potentials. Summary. Generation of an action potential. A threshold stimulus opens voltage-gated sodium channels. Sodium ions diffuse into the axon, depolarizing to plus 30 millivolts. Voltage-gated sodium channels close, and voltage-gated potassium channels close. Potassium ions diffuse out of the axon, repolarizing into negative ions. Potential hyperpolarizes. Voltage-gated potassium channels close, and membrane returns to resting state of minus 70 millivolts.
All right, is that helpful? Okay, so again, for the action potential, I've gone over some basics here. Let me see how many slides we got left. Okay. Let's see. So I have resting membrane potential, also called polarized, 6 things add up to minus 70, but that's an example only. Those 6 things are listed on slides 34 and 35. Action potential, also called the spike potential, peaks at plus 30 millivolts. Again, that varies. Now, be sure you understand this is millivolts, all right? Millivolts. First, you must reach the threshold, minus 55, and again, that's an example only. That starts a positive feedback loop for sodium channels with sodium rushing in. Sequence is on the middle board. Now we got to talk about graded potentials. And graded potentials, this is going to come handy when we talk about graded potentials. Okay, got to be as natural as breathing. "20 minutes, let's see if I could do it. Okay, now we're talking about a third potential, and these are called graded potentials. They are different from action potentials in a lot of ways, and on the bottom of page 22 is showing you the contrast between action potentials and graded potentials. Bottom page 22 of your lecture packet. They are not equivalent to action potentials. They may lead to action potentials, but not necessarily. For one thing, action potentials are what are referred to as self-propagating. That means, I mean, there are some neurons that are really long. The axon can be 3 feet in your body. One cell. I mean, the sartorius is your largest or longest skeletal muscle fibers, and those are many cells fused together. This is one cell, and it could be 3 feet long. What self-propagating means for action potentials is the size of the spike at the beginning is exactly the same size at the end. It does not diminish over those 3 feet. Self-propagating. Okay, I'm putting that on the whiteboard too in blue. Graded potentials are localized, and that means they fade over time or over distance. Action potentials do not fade. It goes to plus 30 at the beginning of that 3 feet. It goes to plus 30 at the end of that 3 feet. It does not fade. But these are localized. And over distance, and this figure here is showing you these arrows are getting smaller and smaller and smaller as it goes away from that beginning of graded potential. Now again, we are going to be concentrating on multipolar neuron physiology. And for multipolar neurons, the dendrites and the cell bodies are the input zones. Okay, this was on the very first slide. Hopefully you guys get that. Textbook shows these arrows getting smaller as it goes away. Now, this is really key, and this is why I drew this up on Monday. For multipolar neurons, the axon hillock is the key. This threshold is all about at the axon hillock. It's got to be minus 55 at the axon hillock for multipolar neurons. Okay. There are 2 different types. There are, I'm just going to change colors here and go red and green because that's what a lot of these textbooks use. You have the EPSPs and the IPSPs. What do they stand for? Well, it's right there on the slide, but what does it mean? Well, it means you've got to know something about multipolar neurons and where they synapse. And that's what I drew this on the whiteboard on that side. That's why I drew 2 neurons yesterday. At the synapse, this before the synapse or presynaptic neuron is releasing neurotransmitter on the postsynaptic neuron. Graded potentials is what's going on the postsynaptic neuron, because those, if you look at the slide, are the middle 2 letters, postsynaptic. The 2 middle letters of these terms is referring to this cell right here. Because the middle 2 letters stand for postsynaptic. Everybody get it? Okay. And if it is depolarizing, boom. If it's depolarizing, it's an EPSP. Because now that postsynaptic neuron is closer to threshold. If it's an IPSP, it's going to be inhibitory hyperpolarizing. You gotta have that be as natural to you as breathing. Before I go over there. So I added that to this. Again, action potentials are self-propagating. Graded potentials are localized. That's why it's in blue. EPSPs are depolarizing. IPSPs are hyperpolarizing. All right. So this is excitatory because it's depolarizing. Middle letter is what's happening on the second neuron. And all those, this is the second neuron right here on the bottom left. How do all those things add up to at the axon hillock? EPSPs are getting closer to threshold. IPSPs are getting further away from threshold. EPSPs are depolarizing. IPSPs are hyperpolarizing. Okay? This is the same thing, and this kind of looks a lot like what I have on the whiteboard right here. Okay? EPSPs get us closer to threshold. All right, so we already covered threshold, and we also mentioned these other topics in that one textbook figure. If it doesn't reach threshold, it's subthreshold, no spikes. If it does reach threshold, you'll get 1 spike. If it is super threshold, you'll get multiple spikes. Now we need to talk about the refractory period, and this is where this after hyperpolarization comes in to play. Okay, there are 2 different types of refractory periods, and this has to do, and right next to the slide, relates to the action potential. These concepts relate to the action potential. Because of the sodium inactivation gates, while a neuron is going through a spike, it's inactivated. In other words, it cannot respond to any stimulus at all, period, absolutely. So we call this first type of refractory period the absolute refractory period. Sodium inactivation gates are closed. They are incapable, no matter how strong the stimulus, of responding during the spike. So that is during the spike itself. Boom. Doesn't matter what stimulus is coming in, it cannot respond. Okay? During the spike itself, look at that graph here and look at the whiteboard in the thumbnail. Does that ring a bell? During the spike itself, this neuron can't respond. Absolute refractory period. Because the sodium inactivation gates are closed. Once it gets back to minus 70, it can respond. But because of the slow potassium gates, it is now further away from threshold. It can respond, but it's going to take a stronger stimulus because it's not at minus 70, it's at minus 80. This is the relative refractory period. It can respond, but it's going to take a stronger stimulus to get the threshold. Relative refractory period. Okay. Now, when you get a really strong stimulus, supra-threshold stimulus, I said you're going to get more spikes. Well, those spikes are going to go boom, 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 boom. Even stronger stimulus, boom, 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 boom. Even stronger stimulus, while eventually you're going to encroach on the relative refractory period that a really strong stimulus can. But once you get up against that line right here, this line in the middle, that's as fast as that neuron can fire another action potential. Because you can't go to the left of this line. Does this make sense? That neuron is maxed out once you approach that middle line. Okay? During the spike itself, absolute. After the spike and during the after hyperpolarization, it can respond. It's just further away from threshold. All right, now let's talk about this concept, and I'm glad I mentioned it. Again, your brains feel nothing. It's just generating illusion, okay? Look at the little thumbnail. How can your brain tell the difference between this and this? Don't ask me to do that again. How can your brain tell the difference if it feels nothing? By intensity coding. And that's what this slide is about, by intensity coding. All those neurons firing action potentials, all the action potentials are identical. How does your brain know the difference between a weak stimulus and a strong one? Well, one of the things is as you are getting stronger stimuli, you're going to fire more spikes. That's why that supra-threshold stimulus should get you more spikes. And this is going to go even up into the relative refractory period. But then once you hit the absolute refractory period line, that's it. Your brain cannot tell the difference of a stronger stimulus for that neuron. Everybody got it? So then your brain or your body's going to recruit more neurons. And that's the second way. And this is why I said this will be key when we talk about recruitment. I thought it was after, but I guess it's today. That's why all neurons have different thresholds. And if you get a really strong stimulus, well, if you get just a normal stimulus, then this neuron's going to fire, minus 55. But if you get a stronger stimulus, not only is this neuron going to fire more spikes, frequency modulation, but other neurons are going to start firing too. That's recruitment. What I would write next to these diamonds. First diamond, it's just about the same number of neurons firing more spikes. Frequency modulation, the same number of neurons firing more spikes. Second diamond is more neurons firing spikes. Everybody see the difference? This is frequency modulation, stronger stimulus, you get more spikes. Everybody see that? That's what those figures should show. Supra-threshold stimulus gets you more spikes. Threshold gets you 1. Supra-threshold should get you more spikes. And if it's really strong, that's going to be really quick. Now this neuron is encroaching on the absolute refractory period. Do you see how these concepts are interrelated? You get this close together, you are encroaching on the absolute refractory period, and now you're going to have to recruit more neurons. Okay? Also referred to as population coding. All right, now let's talk about the postsynaptic cell or neuron. Because the reality is, there are several neurons communicating with this postsynaptic neuron. My purple's finally starting to get dark. Okay. Well, the way this postsynaptic neuron is knowing that there's a stronger stimuli and how it's going to reach threshold more quickly is by this process of summation. And there's 2 different types of summation as well. If it is from different synapses, it's this first diamond. Write that next to this first diamond. Spatial summation is from different synapses. And if they're all EPSPs, boom, you're going to reach threshold quickly. But different synapses are coming in different spots or spaces. That's why it's called spatial summation. Second diamond, just write 1 synapse. First diamond, many synapses. Second diamond, 1 synapse, but it's firing over and over and over again. And it can stepwise help the cell get to threshold by its axon hillock. And with this one, this is where this concept of frequency modulation comes into play. This single synapse has got to fire again and again and again and again and again to get this postsynaptic cell to threshold. Okay. So this first diamond, several synapses, like what I have on the whiteboard, from different spots or spaces, spatial summation. If it's a single synapse firing again and again and again, it's firing again and again and again over time. And the word for time is temporal. That's temporal summation. Okay. Different spots. A, B, and C. You need A, B, and C firing. That's spatial summation. But if it's just A, it's got to fire boom, 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 fast enough to get you temporal summation. So again, temporal summation is only 1 synapse firing again and again and again. Spatial summation is different synapses firing at the same time. Oh my God, how much do I have on this? I'm at 12:20. Let's go ahead and remind me I need to go over the last 2 slides from this week after lecture exam 1 next week. Let me show you the whiteboards here. I'm going to just end this. Yeah, it's already there. Again, over there on the far right is for Wednesday lab. If you're LAP, that's when you log into the Zoom meeting. It is not the regular Wednesday lab meeting. It is the Zoom meeting for Lab Quiz 1. You got to log in at that time. That's the time that the quiz will be available on Canvas for LAP. For everyone else, that's when you log into the Zoom meeting specific for Lab Quiz 1. and when it's going to be available on Canvas. And then as soon as we're done with that, then we reconvene to go and do lab 3. Again, this is showing you these terms postsynaptic, after the synapse, and how those synapses are having effects that are either taking you closer to threshold or further away from threshold. Here's showing you the sequence of the action potential. And again, well, since we are really concentrating on multipolar neuron physiology, we are talking about all the effects of these synapses and graded potentials at the axon hillock. Summation is about how all those signals come together at the axon hillock. Okay. And then again, here's the membrane potentials that we're talking about today. So brought up membrane potentials. We talked about resting membrane potential, action potential, and graded potentials. Receptor or generated potentials is actually a form of graded potentials. So I think I'll put this on the whiteboard after lecture exam 1. Again, maybe not with all those details and colors, but I will include receptor potentials under graded potentials. So you have that all organized. Any questions? Okay, LAP Wednesday students, I will see you actually pretty quickly here. So I'm signing off.